Hi, I'm Simone Flowers, and today we're going to be talking about enzyme kinetics because I got 99 problems, but substrate binding ain't one. So, um, on the first slide, I have a picture of what an enzyme looks like, and the two different colors is basically the two different ribosomal units. And as you can see, there's these beautiful little alpha helices and the beta sheets. So I just wanted to give you kind of structure of what it would look like. Um, so basically, what is enzyme kinetics? Enzyme kinetics is the study of binding affinities of substrates and inhibitors and the maximal catalytic rates that can be achieved. And we really want to study enzymes because maybe we can find a biofuel that is more efficient, that isn't a fossil fuel, that is renewable. And basically what we really want to make sure we learn how to do today is to perform time course experiments and from that make a standard curve, and we want to be able to determine the Km and Vmax values of a certain enzyme. So I believed that the cellulose and the celluliase would make a high Vmax and a low Km value. Um, so just a little preview of the lab. We're just going to be talking about the enzyme celluliase, and we're going to be studying the reaction rates of this enzyme. So background information, what is an enzyme? Well, an enzyme is a biological catalyst, and enzymes lower the activation energy of a particular reaction. They can do this because they have a high affinity for this transition state. So basically what happens is the substrate binds to the active site, it makes an induced fit, and this will lower the activation energy, and basically the enzyme assists in the reaction so less energy is needed. And so this makes it so the reaction can occur more easily and speeds up the rate of reaction. And we're going to be studying this today. As you can see, this is kind of just like a picture of the induced fit in like a little tiny like animation side just to kind of get it clear. But this is how it would actually look. And so here's our substrate. This is the enzyme. This is the active site, and how you can see the shape actually changes once the substrate is binded to the active site and has the induced fit. So what we're going to be looking at today is cellulose, which is a potential biofuel. And cellulose is a component of plant walls. And why we're looking at this is that it can be broken down into glucose, which can be made into ethanol through fermentation. And ethanol can be incorporated into the fuel. So basically, that's why it's so important, because we want to try and see if we can replace fossil fuels. So cellulose starts off here, and then it goes under multiple reactions with enzymes of endocellulase and then exocellulase. But what we're going to be looking at today is the cellulose undergoing a reaction to glucose using the celluliase enzyme. So we're going to be looking at the product being made of glucose from cellulose. However, looking at the product visibly, you can't see it. It's clear, so we can't like visually see how much is actually being made. So what we use is a substrate analog, and we're going to be using p-nitrophenyl glucopyranoside. And basically, it produces this substrate analog will produce both glucose and p-nitrophenyl. And p-nitrophenyl is yellow. So the more yellow being made, the more yellow the product is in the microplate, the more glucose that is being produced. So that's what we're going to be looking at today. We can analyze the enzyme by looking at the Michaelis-Menten kinetics graph. And so it has the reaction rate and substrate concentration on the axis, and it shows us the Vmax and Km values. And Vmax is the maximum rate of reaction when all enzyme active sites are saturated with substrate. So basically that means that when all the enzymes have a substrate attached to it, you can't go under like more reactions. Like all the enzymes have a substrate already binded to it, so you can't do like more reactions. And Km is the substrate concentration that gives half maximal velocity. Km measures the affinity an enzyme has for its substrate. So a lower Km means that less of the substrate is required to reach half of the max. So basically, this all shows the function of the enzyme concentration and enzyme substrate interaction affinity. However, we can't find the Vmax and the Km values directly through the Michael-Menten kinetics plot, so we use a line weaver plot to use like linear data to accurately determine Km and Vmax by basically just inverting the values. This slide, so we use the standard curve for p-nitrophenol product, and we compare these results to the amount of product generated from the cellular bias enzymatic reactions. So from the previous slide, we have the time courses. So basically, we use our standard curve right here that we produce to find the concentrations of product being made from the time courses. So in order to find the concentration values, we wanted to use nanomolar. So here's my conversions of millimolar to nanomole. And these are the absorbance values for my product curve. And then these are the time course readings of the absorbance values. So basically, we converted these absorbance values from the standard curve that I'm going to show you in the next slide into the concentrations that we found. So these are now the concentrations of product being made. So here's a picture of the microplate we used, and as you can see, there's a lot of yellow down here. So obviously from the previous slide I showed you, or 2 slides ago, the diagram. So this was the product standard curve microplate, and then so from here to here is the time course intervals. So basically, as you can see, as you move down, there's a higher amount of yellow in the plate in the little tube, and that's basically just because there's more product being made. Okay. So here is the p-nitrophenol standard curve of the product standard curve. So from this equation, we can use this equation to find the concentration or to find the amount of product being made from the time course intervals. So the y would act as the absorbance value and the x is the amount being made. So basically we do like a reverse equation, and so from, like I said in the previous slide, from that equation we can find the product being made of each of the time courses, and then we can graph them with product amount on an axis and time on an axis, and using the slope of each of the different concentrations of substrate, we can find the reaction rate. So this is the reaction rate for 5 millimolar, if that makes sense. And as you can see, it makes sense. The more substrate you have, the more product that's being made. So as you can see, this is the 5 millimolar, this is the 2 millimolar, and so on. And it decreases as the substrate concentration decreases. So all of the slopes of each of the equations produced from the substrate concentrations of the time course intervals, we can use these slopes in the next slide, and we plug that into the Michaelis-Menten plot. So basically this is the substrate concentration, and then this is the rate, and from this we can put rate on the y-axis and substrate concentration on the x-axis, and this basically shows us how I showed you, or the Michaelis-Menten plot. So we plot this, and as I said earlier in slides, you can't find the Km and Vmax values directly. So what we do, we use the Lineweaver-Burke plot to find the Km and Vmax values. So basically what I did is you do the inverse of the substrate concentration, inverse of the rate, and then you can find, using this equation, the Vmax and Km values, which I have right here. So we were able to determine the Vmax and Km values from the previous graph, and we found that the Km was really high as well as the Vmax. So basically, an equation with a high Km is not good, and that indicates that the enzyme does not bind efficiently with the substrate. So it takes a lot longer for the Vmax value to occur, and that's not what we want. We want it to have a low Km so it can bind to the enzyme. That means it basically binds to the enzyme binds to the substrate a lot more efficiently. I expected there to be a high affinity to p-nitrophenyl glucopyranoside, but that's not the case. Hopefully, what occurred is that p-nitrophenyl glucopyranoside, the substrate analog, makes it so there's a lower affinity. I think the case is that cellulobiose on its own has a high affinity, obviously, for cellulobiase, so it produces glucose at a faster rate. That's hopefully my hope. We'll have to do more tests, and we'll have to do a test that makes it so we don't have to visibly see how much is being made. We can just use another way to find how much product is being made from the enzyme. But that's it. That's all I have for you guys about enzyme kinetics and the lab we performed. Next slide. This is my references that I use. Thank you so much for watching. Thank you.